of tonight's session. So without further ado, let me kind of get the ball rolling here for Dr. Stoker. I'm very privileged and honored to introduce Dr. Stoker. She's a senior research scientist with NASA Ames and is one of the co-founders of the Mars Society. Her recent work is focused on developing instruments for robotic systems for space exploration and testing them out in analog environments here on Earth. She's led field experiments in a number of extreme environments to simulate the Mars environment there, including more recently drilling systems for simulating or for testing for the Martian environment as part of the ARIDS project in Atacama, Chile. Dr. Stoker, we've been hoping to get you in one of our sessions for a while, so we are super excited to have you here. Thank you so much for joining us tonight and kind of sharing your wisdom with us. The floor is yours. I don't know if you've had slides or if you just want to kind of just speak to the audience, and we've carved out time so you can go at whatever pace you're comfortable with. Just let us know if you want like folks to hold Q&A to the end or how you'd like to do that.
Okay. Yeah, my preference would be hold Q&A to the end. I'm going to start my video here.
Okay.
Just so, hi there. So you guys can see what I look like. But I'm going to keep it off for my talk because to save bandwidth. Okay, so I'm going to hit share screen here, and then I think that's going to give you actually my screen. Now I'm going to hit slideshow. Oh no. No, don't do that to me. I've got a new computer and it's giving me weird things. All right, now I think I have a slideshow, except it's not starting at the beginning. All right, here we go. All right, so I'm going to talk to you about not so much how to search for life on Mars, but where the interesting targets are for searching for life on Mars. And you might think that NASA has been actively searching for life on Mars for decades, but as a matter of fact, there really are only 2 missions that have ever searched for life on Mars as we think of, as you would probably think of it, which is to say living organisms as opposed to fossils or some evidence of ancient life. And those were the 2 Viking missions, which are circled on this map in green. And what this map shows is the position of all the landing sites that have ever been visited on Mars or attempted to be visited, because not all of them were successful. So the ones that are kind of more gray were unsuccessful. But the Viking 1 and 2 landers were the only missions that were successful that ever attempted to search for a signature of extant life on Mars. And these missions were in 1976. And this picture shows the young Carl Sagan standing in front of a mock-up of the Viking lander in Death Valley, California. 1976 was the only mission that ever looked for extant life on Mars. It was the first successful landing on Mars, as a matter of fact. And there were 2 identical landers. One was landed at a low latitude and the other at a relatively high latitude, like 57 north, kind of Canada-like latitudes. The landers were focused on life detection. There were 3 life detection instruments, and the 2 landers were identical, so they carried identical payloads. And each one had 3 life detection instruments and an organic analysis instrument. The life detection instruments were based on 1970s technology, of course. So they were invented from first principles to look for evidence of metabolism of soil bacteria via culturing methods. And I don't know why, but suddenly I have a frog in my throat. So carrying on. The Viking biology experiments, again, were attempting to culture soil microbes by exposing them to a nutrient broth and looking for evidence of metabolism. And there were 3 instruments, each of which had a different strategy for doing this search, but all of which basically were culturing experiments. And what happened was, as soon as the experiments were started, as soon as the nutrient broth was introduced into the sample that was put into each of the instruments, there was a reaction. And the reaction was very fast. And it was somewhat like was predicted to occur if there were, in fact, soil microorganisms in play, but different in detail. In particular, the rapid reaction was something you would not expect in a terrestrial environment. And then there was a control experiment, which first preheated the soil prior to exposing it or putting it into the instruments. And in that case, 2 of the experiments still saw a reaction. The 3rd one did not. Now, the control experiment was designed to test the idea that maybe there was some reactant in there that wasn't life. And the heating was designed to kill any organisms that would be in the soil before exposing it and putting it into the instruments. One of the instruments actually did not see a reaction after the heating. So it was giving results that were fully consistent with a biological reaction. However, there was a 4th instrument that was actually not part of the biology package, but was just looking for bulk organics on Mars. This is the gas chromatograph mass spectrometer. And this instrument showed no organics in the soil at levels of parts per billion. And this was thought at the time to be kind of a rule out biology, because biology is composed of organic molecules. And if you had active bacteria, you should also have a lot of dead bacteria around. And those dead bacteria would be seen as organic carbon. So this was, again, it's thought to rule out biology, but in fact, it was a false negative result. And I'll talk a little bit about that, more about that in a few slides. But in any case, the final reports from that mission stated that reactions were more likely to be chemical than biological. And unfortunately, that more likely to be chemical was later interpreted, as is often the case with science, that people don't read all the details and they just read the bottom line. And it was later interpreted to mean there was no life on Mars, which was definitely a overinterpretation of what Viking saw. So I wanted to mention the actual developers of the Viking label release experiment. And this was the experiment that was fully consistent with the biological explanation. And in the last year, a little over a year, both of these investigators have passed away, very sadly, but they were both in their 90s. And in particular, Gil Levin, the one on the right, to his dying day, insisted that he had discovered life on Mars with his instrument. That may not be true, but Gil was unpersuadable that he did not find life. So part of the interesting backstory on all this. So that is pretty much where things stood between 1977 or so and really just before the pandemic took us all down into virtual everything, where there was a meeting in Carlsbad, New Mexico that was called Mars Extant Life, What's Next? And it was a workshop to sort of re-raise the flag about finding extant life on Mars and look at what the possible environments might be where you could search. And I was at that meeting, and this slide here shows an example of what those potential environments might be. So they really are ice, and deposits of ice are found at high latitudes and also mid latitudes on Mars. Salt deposits as a possible site, some of which are ancient and others of which are actually actively occurring even today. And then caves, lava tube caves in particular, have been identified in a lot of places on Mars. And there was a group at this meeting who believed that more habitable conditions can exist subsurface in these caves. And then finally, there's some evidence of deep subsurface liquid water aquifers on Mars. And if they are really there, they probably are relic from ancient Mars and could very plausibly be a habitat for life. So I'm going to talk about each one of these types of environments one at a time and explain both what the case is and what you might look for. So first off, salts. The picture on the top of this slide is showing a map, and the white squares on the map are places where salt deposits, in particular chloride salt deposits, have been mapped on Mars. And it's overlaid on, they're overlaid on a color-coded elevation map. So the colors in the bottom scale go from -8 kilometers to +20 kilometers. We don't see anything or much on Mars that's quite that high, but the Tharsis volcanoes are. And the deepest place on Mars is actually the Hellas Basin. So this is a big impact crater, very big impact crater that's very deep. But all these white spots here are locations of salt deposits. And the reason these are interesting is that large playa deposits in one of the driest, if not the driest place on Earth, in the Atacama Desert in Chile, host microorganisms that live in the salt. And they derive moisture from atmospheric humidity via a process called deliquescence. And I'm going to explain deliquescence to you in another couple of slides, so hold that thought. On the right side shows a crystal of salt with a red or orange pigment inside of it. And this is actually a pigment caused by a viable microbial colony. And viable microbes can actually live inside salt crystals, living in fluid inclusions inside salt crystals for up to 100 million years. So that these salt deposits are potentially interesting as providing signatures of life, not necessarily from modern times, because even 100 million years on Mars, long since Mars has been a warm and wet habitable sort of place. Okay, I was part of a mission in 2008 called the Phoenix mission. It wasn't named after the Phoenix, Arizona town, even though it was operated in Tucson, Arizona. It was named after the Phoenix bird, the thing that rises from the ashes and flies again. So the Phoenix mission was actually a reflight of a mission that failed in 2001 and carried much of the same payload as that mission that failed. And that was why it was called Phoenix. But anyway, it landed on the northern plains ground ice near the North Pole, 68 degrees north, above the Arctic Circle. And it was actually landed there to assess the habitability of that ground ice, which at the time was really the only certainly known ground ice. But its major discovery was that 1% of the soil in that location, and I actually subsequently realized pretty much everywhere on Mars, 1% of the soil is this unusual salt called calcium, magnesium, or sodium perchlorate. And this is the chemical formula of this salt. So it's 4 chlorine, a chlorine and 4 oxygens and a whole lot of water molecules attached to it. So why is this particular salt important? Well, it burns organic carbon. So literally, if you heat perchlorate up with organic carbon, it will burn, it will combust. Those oxygens will become available like like flame, and you literally will oxidize the organic carbon very rapidly. And the GCMS instrument, this was the instrument that detected no organic carbon on Mars, actually took the soil in and heated it up prior to making its analysis. This is how these instruments work. And it's very likely that the presence of perchlorate, when combined with organic carbon and heated up, caused the oxidation of the organic carbon to carbon dioxide. And that actually has been observed in subsequent missions as well. Another thing about perchlorate is it's antifreeze. At its eutectic point, the freezing point of liquid solution is -75°C. And this is the temperature of the subsurface ice at the Phoenix landing site, as a matter of fact. It's liquid at extremely low temperatures. And finally, it's very strongly deliquescent. The perchlorate salt crystals grab water out of the atmosphere at relative humidities of like 80%, and they will then remain liquid down to about 20% relative humidity. So they are have unusual characteristics that might have a lot of implications for what we're seeing on Mars. So next slide shows again, a map showing where deliquescence of perchlorate is possible on current Mars. And the upper section of this picture shows the water activity of that perchlorate. And the lower picture shows the temperature. So water activity is defined as the vapor pressure over the solution over the vapor pressure of pure liquid water. It is a measure of the availability of water to life. It's used as that. In all of what we know about the capabilities of terrestrial life, nothing can survive, bacteria specifically, cannot survive or thrive. Actually, they can survive, but they can't grow at a water activity less than 0.6. They also can't grow at a temperature less than -25°C. Well, the bottom picture here shows what the temperatures on Mars are, when this deliquescence is occurring. And the thing that drives deliquescence to occur is the fact that the relative humidity has to be high enough for the water to get pulled into the salt crystals. And on Mars, current Mars, this only happens at night. And the reason it happens at night is because the temperatures drop very low, down to like -75 centigrade. And when the temperatures drop that low, it's like when you get frost on the ground when it gets really cold. So basically, the relative humidity is high when the temperature is very low. So, but the point is that there is liquid water occurring on Mars, or very likely is liquid water occurring on Mars, in very large areas of Mars in modern times. It's very salty water, but it's there, and it's really there at night. Probably nobody can grow in it that we know of, but nevertheless, it's an interesting thing to think about. Okay, so the next thing I wanted to talk about is ground ice. At the time of the Phoenix mission, and again, this was 2008, it was really thought that ground ice was a relatively rare phenomenon and it was confined to very high latitudes. But subsequent analysis, and the analysis that's shown in this map, that was actually quite recent paper, published by a group of authors led by a fellow named Picot, shows that ground ice is actually widespread at all latitudes north of about 40 degrees. And over here on the right, again, this is a color-coded scale showing you the depth to this ice table based on this analysis. And what is showing is that over very substantial parts of Mars, there's ground ice within a meter of the surface. And actually in many places, much closer to the surface than a meter. Now the white squares on this map are actually places where meteors have hit the surface and excavated into that ground ice. And on the right is a picture from the HiRISE instrument. This is an orbital mission that has this instrument. And this shows basically this white splotch, which occurred after this meteor hit the surface. And HiRISE is repeatedly imaging the entire surface of Mars at very high resolution to look for evidence of things like things that are changing in real time. And specifically the meteors hitting the surface has become a quite active area of research. So the white places are places where white ice was excavated. The red dots are places where meteors impacted, but white ice was not excavated. And these black regions are areas that have a lot of dust. And in fact, they did not perform this analysis on these areas because they were too dusty. So they actually might still have ice, but they can't be, or it could not be observed using the method that was used to make this map. Okay, moving on to talk about why is all this ice there. So basically changes in the obliquity of Mars drives climate change. It drives it on Earth as well. But in the case of Mars, the obliquity changes over very, very wide range over a relatively short period of time. So currently the obliquity of Mars is about 25 degrees. But 5 million years ago, it was 45 degrees. And this is a plot. This picture here in the middle is a plot of the obliquity of Mars going back 10 million years. And here we are today at this 25. And what you can see is that the obliquity is going up and down very rapidly, but staying in a sort of under 35-degree obliquity up until about 4 million years ago, when suddenly it jumps. And above or back in time more than 5 million years ago, the mean obliquity was like 35 degrees. And the maximum excursions were 45 and even up to 50 if you go further back in time. Well, what happens is that when the tilt of the Mars axis changes like this, the amount of insolation falling on a particular place on the surface of Mars changes along with it. And I've plotted on the right the summer solstice insolation at the Phoenix site along with the obliquity. And what you see is that when you get up to these high obliquities, the amount of sunlight falling on the surface at summer solstice is more than twice as high as it is right now. So this actually has a very big impact on the temperature of the surface and what happens to the ice at the surface. So the next picture here shows that climate models show where pure liquid water is thermodynamically stable on the current obliquity of Mars. So these are number of days a year that liquid water could exist on Mars just thermodynamically, meaning the pressure and temperature conditions would allow pure liquid water to form. And so basically it's all between the equator and 45 degrees where you could currently form liquid water on the surface of Mars. But what happens when the obliquity is 45 degrees? And that's shown in the next slide. So now what happens is that the locations where pure liquid water can form move north, and they the region north of 45 degrees latitude actually has many days a year that pure liquid water could form. So what this is showing basically is just the thermal properties and the thermal impact of having obliquity change on Mars. So effectively what it means is that as obliquity changes, the locations of ground ice also move around. So the ground heats up, water vapor goes into the atmosphere, and it moves to other locations, which would be colder and kind of act like a cold finger to capture that water vapor. So the next slide I'm showing is the ice table depth from a recent paper, very recent paper by Mike Mellon and Hannah Sizemore. And the bottom of this slide is showing obliquity over the last 2.5 million years. So this is even more recently than this big excursion to a very high obliquity, mean obliquity. But what it's showing is that even in the last 2 million years, as the obliquity goes up and down, for the different landing sites on Mars, let me try to explain this again. So what this is showing is the depth of the ice table that this climate model predicts as the ice moves from the northern plains into the mid latitudes, and it's showing what the ice table would be at different times in the past for the different landing sites that we have visited or plan to visit. Rosalind Franklin is the ExoMars rover that is planned to be launched next summer. So Perseverance is this red line. Curiosity is the green line. So Curiosity is Gale Crater. This is the lander that landed in 2010 and has been driving around on Mars for the last 10 years. What you can see is when you get back to just 500,000 years ago, it's 500,000 years ago, the ice table at the Curiosity site is only like a few centimeters beneath the surface. So it's, the size of your thumb beneath the surface. And the Zhurong rover, this is the Chinese rover. It's actually at a higher latitude. It's even, the ice table is even less far under the surface. Now, this is not where it is now. This is where it is 5 million years ago. So the fact that at zero, this is now, none of these landing sites have ice near the surface in the top 10 meters of the surface, actually. But 5 million years ago, or 500,000 years ago, many of them did. The reason this is important is that this emplacement of ice basically is unstable, but while it's moving, or while it gets heated up so that it can move, it's potentially habitable. It's habitable ice. So a less dramatic understanding of what is happening with the ice table was published like 10 years earlier by a fellow named Aaron Zent. He's actually a colleague of mine at NASA Ames. And he was looking specifically at the Phoenix landing site and what happens to ground ice at the Phoenix landing site as the obliquity changes. So his models showed that at high obliquity, at 45 degrees obliquity, the ground ice at the Phoenix landing site hadn't moved. It was still there, but it was warm. And it was in fact warm enough for microbes to grow in it at down to depths of almost a meter. And for long enough for microbes to grow. So this is like a time scale for like in terms of fractions of a year. So this is like a tenth of a year. The ground ice is warm enough for microbes to actually grow, not just survive, but metabolize and make more microbes. Okay, so that's kind of the ground ice story, which I think is the most important story from the point of view of human exploration, because the human missions would actually like to land on mid-latitude ground ice and extract it as a resource to make rocket fuel and breathable air and other commodities that are useful for humans. So the next target that I wanted to talk about is caves. Now, the reason we know there are caves on Mars is that, again, from high-rise imaging, we see places on the surface of Mars that appear to be skylights. There are places that there's a dark region where you can't see into the subsurface. Maybe I should stop right now and just make sure you guys are still there, because I'm seeing what appears to be a frozen screen.
Hi, Dr. Stoker. No, we're definitely still here. And
okay.
Yeah, no, and this is.
And you're keeping up with me.
Trying.
Okay.
Some folks maybe a little better.
Okay, good.
Yeah, no, absolutely. This is terrific.
All right. Okay, so on the left here is a section of a map, or really an image of the surface of Mars, showing these green squares, which are the locations of hundreds, or actually 1,000, hundreds of caves or lava tube entrances that have been mapped on Mars. This map from Cushing et al. In 2015 shows or has mapped the locations of over 1,000 cave entrances. Most of the ones are associated with Tharsis volcanism. These are these big volcanoes on Mars, but some are in other locations as well. The reason that caves are interesting is that on Earth, they are basically windows into the subsurface, where the subsurface is warmer and can actually host habitable conditions. Many caves on Earth are actually wet and have a lot of water in them, and a lot of features carved by water or formed by dripping water. So some of my colleagues are passionate studiers of caves. And what they find in caves are very morphologically unusual types of microbes that basically live on rock in the caves. So, and they themselves will look very much like rocks, or like coatings on the rock that might be a different color, but still are hard and rock-like in nature. So Diana Northrup, a colleague of mine who studies these, calls them microbes masquerading as minerals. But if you scrape a sample off of these microbial colonies and you look at them under a scanning electron microscope, you see in many cases very unusual morphologies that are absolutely biological in nature. There's no way they can be mineral, but they are nothing like any microbes that live in soil, for example. So recently, of course, we have the opportunity to explore Mars with flying devices. I was very excited to see in the Perseverance mission the addition of the Ingenuity helicopter. And the reason that the drones are important as a potential life detection technology, not that they can themselves detect life, but they have the ability to fly down into a lava tube entrance, down a skylight, and be able to actually just do a temperature and humidity measurement would tell you right away if that was a habitable environment. Now, getting actually down to the surface and sampling for life would be a much more challenging problem, but at least determining the habitability with a helicopter would be a very interesting type of mission. And I'd be surprised if the next time that we have a competition for Mars missions, I'd be very surprised if there isn't another helicopter included in that competition. Okay, so the next slide here is another map. And this map is showing the locations of caves and ground ice less than a meter deep. And this particular image was part of a Planetary Protection Committee report. The Planetary Protection Committee is actually a National Academy of Sciences organized committee, reports to the Committee on Space Research, which is an international group that has the charter to govern whether or to govern planetary protection. They are in charge of that planetary protection, meaning contamination, either forward or reverse. Contamination of Mars by terrestrial microbes or contamination of Earth by Martian microbes. Interestingly, this committee has recently met and they have gotten interested in this whole question of caves and ground ice. And this map is showing the colors which are not the sort of light purple are places where there is 2 lines of evidence that indicate the presence of water ice. The purple areas, there's only 1 line of evidence, and this is the presence of high percentage of water equivalent hydrogen. However, those areas with the high percentage of hydrogen measured by a neutron spectrometer are not necessarily thought to be ice rich. So think of the other colors as kind of guaranteed to be ice rich, and the purple colors is less likely to be ice rich, but nevertheless have a high percentage of hydrogen in the soil, possibly hydrated minerals. And what you see is in these black dots, this is where all the caves are. So there are places where there's both ice and caves. And that's actually, again, very interesting because the presence of caves and ice together kind of acts as a logical study area to look for evidence of extant life, because this is a high probability of a sheltered, warm, quite possibly water-rich place. Okay, so finally, there's a 4th area, which is deep subsurface water. In 2020, there was a paper in Nature that was published showing evidence of from a ground penetrating radar. This is an orbital ground penetrating radar that has been flying for a number of years. And it was actually a reinterpretation of the data from this mission called Marsus, or from the radar called Marsus, which was interpreted in terms of there being lakes under the South Polar cap, or South Polar layered deposits. The picture on the lower left is actually from their Nature paper showing this presence of lakes. However, since this paper, there have been a number of other papers that have refuted it, or said this can't possibly be liquid water. So the arguments against it being liquid water is that it's too cold for even any conceivable known brine to be liquid at those depths. And subsequent papers have found that there's actually a lot of these spots, not just the ones that were published in the original Nature paper. And some of them are at shallower depths. And they're radar bright, meaning that they are highly reflective in radar, which means that they have a high dielectric coefficient. But it has been argued that there are other ways to make a radar bright area other than ice, or other than liquid water. It could be conductive ice or even clays. So that's still under consideration. So, so finally, what does this all have to do with human exploration? Well, going back to Viking, the general view has been that the surface of Mars does not host life, and that if there is life on Mars, it's unlikely that humans will interact with it. It's that you can actually pick places on Mars to land humans where there's no risk of inadvertently encountering life. But the evidence is not very good. It's only one set of instruments that's ever actually tried to determine if there's life on Mars, going back to Viking. And this probably doesn't constitute due diligence with respect to returning samples of Mars to Earth, in particular astronauts that have been encountered Martian materials. And it turns out that it's actually required by the Outer Space Treaty that you don't bring samples back to Earth, that there's a risk of contaminating Earth with those samples. And in fact, the Perseverance mission is the first step in a plan, a robotic mission to return samples to Earth. And it has taken very elaborate precautions to make sure that those samples are contained and don't, contaminate Earth. Human missions, it's impossible to do that. So I would argue that in situ life detection mission is required to go to the actual landing site or a landing site, a candidate landing site that you would send humans to do a life detection mission to verify that there is no life. So in conclusion, it's been 45 years since Viking. Isn't it time we resume the search for extant life on Mars? Carl Sagan says it, I say it, and there's 4 types of environments you could search in, but the most important thing to do is to actually look, because we haven't looked since 1976. And that's it. With that, I can take a few questions.
Dr. Stoker, I thought to say that's it. I mean, that's a heck of a lot. This is awesome. Thank you again so much for this. We did have a bunch of questions come in here. I'm going to scroll up and I'm just going to kind of go through them and Dr. Stoker will take your lead and we'll go through however many we go through. And I think I've got a couple that came in beforehand as well that I've got an email somewhere as well. So, just starting at the top, this is from Kent. Has there been any recent research on the unique conditions in Hellas Basin?
That's a real good question. I think Hellas is a fascinating target and a very understudied target. It's actually a reason. There's a reason that Hellas doesn't get a lot of attention, and it has to do with the fact that it's always covered with clouds, or frequently covered with clouds. So there are very few studies of the Hellas Basin, but it is the deepest and undoubtedly the warmest place on Mars, because it is deep in the southern hemisphere, where currently the southern summer occurs at perihelion, and therefore, summer in the southern hemisphere is actually would be more warm than in the northern hemisphere, except that the southern hemisphere in general is much, much higher than the northern hemisphere. And Hellas is the exception because it's a very deep crater. So that's about all I have to say about Hellas.
So you're not aware of any particular recent research, or what about any mission plans that you've heard kicked around?
No one has proposed a mission to Hellas that I am aware of.
Okay. Interesting. Okay. Another one, I think actually Mike suggests this one. What have your field studies in Antarctica and the Arctic taught you about the options for searching for life on Mars? And I know I've seen you make kind of other statements in terms of like the advantages of like when to use robotic versus or where to use robotic versus where not to use it. What do you think about some of the options for searching for life from a technical perspective?
Well, so there are sort of two questions there. One is, what do we learn from studying the polar regions of the Earth? So really, since the advent of the NASA astrobiology program, which ironically was started as a result of the claim made by Dave McKay and colleagues that there was evidence for biosignatures in meteorites that came from Mars. And those meteorites actually were found in the Earth's south polar region. But there are a lot of researchers who study life in the Antarctic. I actually have not been one of those researchers, but there are certainly people who do. And what has primarily been learned from that research is the extremes of life. The temperatures at which metabolism has been observed have been extended as a result of that type of research. Also, the other kinds of extremes. But there are certainly places in the Antarctic which appear to be devoid of current life and current metabolism. And most of the researchers that I know who study life in the polar environments really don't think there's life on Mars, because they think that Mars is enough worse than its terrestrial analogs, and that life is really just barely hanging on in the polar environment. So it's the temperature that seems to be the limiting factor, not the presence of ice or the presence of liquids. It's just the cold temperature itself.
Interesting. Now, did I understand correctly from one of your slides that there are regions where at certain depths, like terrestrial life could survive today if kind of transplanted there? Is that the implication of one of the things that you said?
Well, we don't know. I mean, what we've spent the last 40 years doing, or really, the last 20 years doing, is driving around looking at rocks and trying to come up with a compelling evidence that early Mars was warm and wet. And I think that case is, really made. I mean, it's a solid case that early Mars was warm and wet. But it was very early Mars, 4 billion plus years ago. And that it seems to have become a cold, dry desert very quickly. And it's actually kind of puzzling why it would have been warm and wet in its early history, because the sun was actually less strong in the early, 4-ish billion years ago. But, our understanding of Mars is evolving, and this understanding of the amount of ground ice there is and how it's moving around is very recent.
Mm-hmm. That's interesting. Next question. I'm going to skip one ahead here for Catherine. She asks, how deep have we been able to drill for core samples?
On Mars?
Yes.
2 centimeters.
2 centimeters.
I mean, the samples that are being drilled right now by the Perseverance rover, those are the core samples that we have drilled. And I think their drill is, maybe I'm underestimating, maybe it's 5 centimeters, but it's very, very shallow. We haven't really scratched the subsurface. Now, the ExoMars rover, which is the European rover that will launch next summer, has a 2-meter drill. And so it is actually capable, should it encounter a place where there's ground ice, it's capable of sampling it. But ground ice is not expected to be present in the location where it's going to land.
Okay, interesting. I believe Ed Eisler, you've raised your hand.
Yes.
Go ahead. Yeah. What's your question?
I believe there has been some modification in the restrictions on where we can look for possible life on Mars. In other words, the restrictions, I believe, prohibited us, NASA, and probably the European Space Agency, from going to places that were most likely to contain evidence of life on Mars. Now, I'm wondering, do these restrictions, the current restrictions, would they apply to the Starship when it goes to Mars in 4 or 6 or 8 years? And would these restrictions also apply to the China's National Space Administration? That's it.
Okay, so let me try to take it in reverse order. So let's China, Starship, and
Okay.
what we're doing now. So the restrictions come from the Planetary Protection Subcommittee, which I mentioned. And so the restrictions are for 2 things. One is to prevent forward contamination of Mars. In other words, contaminating Mars with things that might be later misinterpreted as Martian, but were brought from Earth. Then there's the question of reverse contamination. This is bringing stuff from Mars back to Earth and contaminating Earth with something we didn't know was there, but caused a big problem on Earth. So you asked about China. I do not know if China is a signer of the Outer Space Treaty. If it is, then they're just as restricted as we are. The second thing is, what are the restrictions? So for robotic missions, the restriction is that the mission, if it goes to certain locations for which by an evaluation of the Planetary Protection Office could actually allow terrestrial organisms to survive, then that spacecraft has to be sterilized, or anything that's going to touch the surface that might be habitable has to be sterilized. Now, the Viking missions actually started that protocol. The Viking missions were sterilized. The entire payloads were actually put in a big oven and heated up. The whole spacecraft. That has not been done ever since Viking, and it's always thought to be too expensive. But Viking did it. The missions that don't want to do that, that don't want to sterilize the thing that's going to touch the potentially habitable environment, are going to places where there is no expectation of there being a potentially habitable environment. So Perseverance is not going to a place where it's expected that there's habitable conditions will be encountered. The ExoMars, I believe, sterilized their drill. And the mission that we proposed, the icebreaker mission, sterilized the drill. The drill would drill into ground ice with the expectation that that was a potentially habitable environment, but the whole mission wasn't sterilized, the whole spacecraft, but just the drill, because that's the only thing that would touch the subsurface. SpaceX and their planned missions, there's no way that they can do a sterile anything. You cannot sterilize human beings, and you cannot sterilize a spacecraft. You could land in a place that is expected to not have a habitable environment. And that's kind of what this whole story is about, that, you know, there are areas that aren't, let me go back to this map. There are areas that aren't colored on this map. And many of those areas are places where we are landing. Most of our missions have landed in these not colored areas. And the reason that we landed there is because of these restrictions. Now you can get around the restrictions if you want to pay the cost of sterilizing your equipment, but you cannot do that with humans. And so SpaceX is not themselves a scientific company, they're an engineering company. And they are kind of expecting, you know, I don't know exactly what they're expecting, but the risk is that the Outer Space Treaty says that you can't bring sample, or you can't bring material back from Mars, and that includes astronauts, that could potentially contaminate Earth. And anyone, I mean, me or you, could file a lawsuit and stop SpaceX from launching. And that is where things stand right now.
Very interesting. That's interesting because that doesn't get much, at least I haven't seen that last point discussed much.
It is not getting discussed at all, which is
Yeah.
actually why I think we need missions to go verify if there's life on Mars or there isn't.
Yeah.
You know, we have kind of been, you know, dancing around this question for a very long time.
It's interesting. All right, we've got, I think, Dr. Stoker, if it's okay, maybe one or two more questions.
Okay.
Okay, so one, also, are you ever any plans by NASA for Ingenuity, maybe sort of as a final hurrah, to actually go down and do a lava tube? Is that within its range, and any thoughts on that?
Well, I think if there were lava tubes around where the Perseverance is located, that they probably like to do it, but don't believe there's any evidence that there is in that area. You can see on this map where the nearest one is from the landing site. It's pretty far away.
Yeah.
You know, that Ingenuity helicopter was a technology demonstrator. It wasn't really intended to be part of the mission. It was it was there to prove you could fly on Mars. But now, of course, that it's been phenomenally successful and it is flying on Mars, they're using it as part of the mission.
Right. Yeah. That's terrific. I want to give kind of a last call to folks online to, if you have questions, please either raise your hand or just type it into chat. I had one about the water cycle, or a presumed water cycle. That, I remember from when my kids were young, they would do science projects about the water cycle on Earth, and it's the various stages that it goes through. So is it as simple that the water cycle, the presumed water cycle on Mars, is basically evaporation and deliquescence? Is that
Well, the water cycle on Mars does not go through rainfall. It doesn't.
Right.
It's too cold and too dry for liquid water to be in the atmosphere. So it's basically sublimation from the ice deposits and deposition in other places. So yeah, I think
Okay.
that's the way you have to think about it.
Okay. All right. Very good. Okay. And I don't see any other questions. Anybody raising their hand? Let me check. I don't see anybody.
Yeah, I think it's time.
Okay.
You guys still have chapter meeting stuff to take care of, and I've now taken up a whole hour, so
No, it's
I
been
probably
fantastic.
better get going.
No, so Dr. Stoker, thank you again so much. This is something we wanted to do for a long time. We were very excited that you came and joined us for this time and shared your thoughts and with us. So thank you again very, very much.